I'm Harold Varmus from the class of 1961, and I want to take a moment just to thank those who've organized these programs. We come to reunions thinking about ourselves and how old we've gotten or haven't gotten, and how old our classmates have gotten or haven't gotten. And it's nice to think a little bit about something outside ourselves. And I think a lot of us have been doing a lot of that. And it's useful to look at how our fields of endeavor have changed, matured, often as the results of accomplishments of those in our generations. So what I'm going to do today is talk about my own field, which is medical science. This is going to be largely celebration, not criticism. I will say some things that I know can be modified and amended to express some concerns. I'm going to talk about health science, not health care. We heard about health care this morning. That's much too tough for me. Health science is a lot easier. Let me begin, before I show you some pretty pictures, by telling you what I hope to achieve. So my goal today is to actually wake us up to what has happened in our own lifetimes, to outline remarkable, indeed revolutionary changes that have happened in biology and medicine in the span of my own career and in the lifetimes of my classmates. Changes that have, for the first time, shown mankind how life works, how certain diseases arise, and how health and health care could be improved through knowledge, both here and abroad. I'm also going to argue that these changes were achieved and were possible in large part because of the kind of ambitions that exist in our country, the political goals that were set, actions that were taken, and the research institutions that we've all created. And this weekend of retrospection is, for me, an opportunity to review and appreciate what we, in my own class and other classes, have witnessed. So here's a synopsis of what I'm going to tell you. Our nation decided to invest heavily in science, including medical science, when my classmates were very young children. In our teenage and college years, this resolve continued to grow, and the underpinnings of modern biology were discovered: the double helix, the central dogma, the genetic code. More on these things in just a moment. Over the next 5 decades or so, knowledge about the major causes of mortality have been increasingly exposed, and these diseases have come under increasing degrees of control. Infectious diseases, cardiovascular disease, with increases in our life expectancy and, importantly, reductions in disabilities, especially in those countries that belong to the advanced economies. Many of these advances against disease have been possible because of progress in genetics, and that's the one aspect of our science that I will emphasize, but also through the development of recombinant DNA technology, the development of new vaccines and drugs, manipulation of the immune system, neurosciences, new technologies, many other things. More recently, some of these improvements in health are being made in the developing world as well, through control of malaria, AIDS, and other infectious diseases. And now increasing attention is being paid to the non-communicable diseases, heart disease, obesity, diabetes, and cancer in those countries where life expectancy is now rising as a result of control of those other diseases. At the same time, the way we do medical science has changed, and largely for the better. Partnerships and teams have become the norm. Sharing of materials, information and internet-based sharing of publications. These have become the norm in practice, and hence have become a standard and hence more common. Science and medicine have also been portrayed in interesting ways, both in fiction and nonfiction. I'll allude to some good examples that you might want to peruse later on during the course of my talk. Yet, despite all this, and perhaps ironically, this is not a time either for comfort or for self-satisfaction in the scientific community. For those of us in the medical arena, the opportunities to build on the revolution in biology has never been greater. Further, the needs to expand medical progress against infectious diseases and diabetes are compelling, and especially against the rising tide of obesity and its consequences. But public support for science remains fickle. Congress has responded to the current economic crisis by cutting budgets for science, even this year, for the first time in my experience at the NIH, where we've seen a 1% decline in our money compared to 2010. The likelihood of success in the grants approval process for new investigators and old is at an all-time low, and our ability to recruit scientists from abroad and attract our own best talent to science have declined. In short, we're at risk of failing to achieve the potential in science and in medicine, and of ceding dominance in those fields to competitors in Europe and Asia, and I'll mention that in a moment as well. So before I can go further, for the benefit of those who are not in my class and don't know me, a personal note. 50 years ago, as I left Amherst College, I would not have been expected to be the person telling these stories. During my time at the college, I abandoned my anticipated medical track for college journalism and graduate school in literature. Then, against odds, I changed course, went to medical school, learned to do science at the NIH as I neared 30, and we were fighting a war in Vietnam that I didn't want to participate in, and then devoted most of my career, I hasten to say, thus far, to medical research, especially to the study of retroviruses and cancer, to the running of some medical institutions, including the NIH, Memorial Sloan Kettering Cancer Center, and now the National Cancer Institute. So I'd like to begin by giving you a brief synopsis of how this country became a dominant power in research in general and medical research in particular. At the time that we were all in diapers, FDR took a special train trip during the campaign for his third presidency and came to the NIH, then just the National Institute of Health, to dedicate an expanded campus on October 30th, 1940. And that campus has grown into about 10 times the size that FDR rededicated, a place where I and many in this room work and have worked over the years, a major source of our influence in the world of medical research. By the end of World War II, FDR and, importantly, his advisor, Vannevar Bush, and Harry Truman as well, began to appreciate the contributions that science had made, science largely funded by foundations and the private sector, to our triumph in World War II. There was penicillin, there was new treatments of malaria, there was an atomic bomb, there was improved radar, and all of these things brought the science advisor to FDR and Vannevar Bush to write a book called The Endless Frontier, which has provided a blueprint that's guided American science for many, many years. And that book laid down a fairly simple precept, namely that federal funding of basic discoveries through investigation of interesting phenomena that might have consequences for the way we conduct the business of the world would lead to discovery, to practical applications of those discoveries, largely carried out in the private sector. When we entered Amherst College in 1957, shortly after we arrived, a major event in the history of the world occurred, namely that the Soviet Union placed an object into space, into orbit around the world. Thank you. And that obviously posed a major challenge to our dominance in science and technology, which only increased our determination to spend more and do more in this sector. One important aspect of our response to that challenge was the declaration by JFK just a few days more than 50 years ago, when he addressed Congress and asked that Congress allow the U.S. to place a person on the moon before 1970. This was an engineering challenge in many ways, but it also galvanized interest in so-called STEM education, as we call it now, science, technology, engineering, and math, and led to marked increases in our investment in many sciences, including the medical sciences. It was also a prototype for other kinds of challenges, and in 1971, Richard Nixon signed the National Cancer Act, which declared a more difficult challenge, and one that required us to learn a lot more through basic discovery, and one that has yet to be completely successful. But as you'll see, as I talk in more detail about cancer, not only did this provide the funding that led to a tremendous expansion of the National Cancer Institute and to the effort against cancer, but led to work on a very broad scale that has informed basic science and clinical science. Let me talk a little bit about how these investments have played out in our efforts to understand the fundamentals of life. When we were still in junior high school, Watson and Crick unveiled a model for DNA. DNA was thought probably, but not necessarily, to be the vehicle by which information was transmitted in all living cells. And their view of this molecule, a double-stranded helix that you've all heard a lot about, composed of 4 kinds of components, the so-called bases or nucleotides, fit the need to have 2 things. One, information contained in that molecule, and that could be acquired by having these bases in a certain order. And secondly, the need to have that information both copied and read out. And that was possible because of what's called base pairing, C's with G's, A's with T's. And these 2 fundamental concepts of what one can achieve through, oops, I don't want that. What do I want here? Didn't really mean for that to happen. Let's just go back. Led to an understanding of what molecules were involved in both replicating the information as DNA gets duplicated and expressing the information in a way that a cell could make use of it by making another nucleic acid called RNA and having the RNA translated to make a very different kind of molecule called protein. This so-called central dogma, DNA makes RNA makes protein, is a fundamental of how all living systems work. And just after our graduation from college, when the Soviet Union decided that Lysenko's ideas were bogus, and they invited the International Congress of Biochemistry to go to Moscow, I traveled there as an incipient graduate student in literature with my pal Art Landy, who won the biology prize at Amherst that year and is now a distinguished professor at Brown. And we heard Marshall Nirenberg, a great NIH scientist who unfortunately died last year, lay out the ground rules for how information is actually encoded in DNA with the 4 building blocks, making words each composed of 3 letters, and each of those triplets telling a cell to insert a certain one of the 20 amino acids in proteins into a protein chain. This, again, a universal truth, basically the same in plants and bacteria and animals. Fundamental precepts that were obviously not known before then. These tools were incredibly important in the rise of the so-called recombinant DNA technology revolution that led to a new ability to take information from one species and express it in the cell of another, an exercise that fundamentally changed the way we do science. It also led to the development of a new industry because you could take genetic information from one organism, like a mammal, and put it into a bacterial cell and make a protein that might be useful for medical care. And Boyer and Cohn, who achieved that, were, of course, richly recognized. And Herb Boyer founded Genentech, which introduced a new concept into biological research, namely the possibility of making large amounts of money and making great products and influencing the way the pharmaceutical companies work. And this is one of the bricks that has created the notion that medical science, the NIH, are engines of economic productivity. Understanding our genes, of course, ultimately involves knowing about and understanding the entire repertoire of chromosomes in our cells. And that led to the Human Genome Project, which also encompassed the analysis of the full sets of genetic information in many, many other species, from bacteria and yeast through a whole panoply of plants and animals. that was accomplished for human beings in 2003 at the cost of nearly $15 billion over 15 years. Sorry, $3 billion over 15 years. That told us a lot about all the sequences, especially those that encoded proteins. That totals about 2% of the entire genome, and the rest remains part of the mystery to be solved. The Human Genome Project also illustrated another concept I want you to learn about how we've changed the way science is done. This was a team effort. 5 very large sequencing centers and many much smaller ones that worked in a collaborative fashion over the course of 15 years to achieve this remarkable outcome. This technology of sequencing human genomes to understand how individuals function and to sequence many other species as well began as a very expensive process. At this point, roughly 2011, one can do what used to cost billions of dollars for about $10,000, and the price will soon be down, in the view of most of us, to below $1,000, a notion which is going to completely transform both science and medicine. This is also a source of international competitiveness, not unlike what happened when Sputnik was launched, namely that other countries are taking the advances that have been made here, building their own technological centers. One in Shenzhen, China, recently visited by Francis Collins, director of NIH, depicted here as a place that now has the technology to sequence 10,000 genomes a year, with technological capacity that may exceed what's present in all of our genome centers combined. So this is a real challenge, and one that we should be responsive to, in my view. Now, science of this sort in the health arena is not done just to learn about how life works, but also to think about how we can better control disease. And it is true for multiple reasons that in the years since we were born, in the late 30s, early 40s, life expectancy has continued to be extended. At the start of the 20th century, life expectancy was 47 years. By the time we were born, it was a little over 60 years. Now it's getting close to 80 for men, a little more for women. There obviously is a lot of heterogeneity in these numbers. If you begin to segregate the population, there are health disparities in our country between rural and urban folks, between among races, among populations that are particularly deprived economically and socially. But on average, life expectancy has been markedly changed. And there are lots of reasons for that, many of them medical, some social. If we begin to look at specific diseases, for example, as you can see, the heart disease that killed my own father and probably the parents of a lot of folks in this room has been reduced by about 67% over the last few decades. These are age-adjusted numbers, and that's attributable to many things: more rapid response to acute cardiac events, to new ways to establish, to prevent the accumulation of plaques in arteries through the use of cholesterol and low-density lipoprotein by the so-called statins, and many other ways to monitor disease. The facts with cancer are a little more complicated. The age-adjusted death rates rose slightly over several years for reasons you'll see in a moment, but have been consistently declining overall by about 1% a year. Numbers with cancers have to be looked at more carefully because cancer is not one disease, a major message I'd like to leave you with. Cancer is lots of different diseases, and we have to look at each one differently, both with respect to epidemiology, to incidence, to death rates, to genetic causes that I'll come to in a moment. For example, most prominently in this chart of mortality rates for men, lung cancer death rates rose dramatically as smoking became very common, and the death rates have come down quite dramatically since roughly 20 years after the Surgeon General issued his report in 1964 and began to persuade people that smoking was bad. Colon and rectal cancer death rates have begun to decline, or down about 40% now as a result of much greater use of colonoscopy and examination of of feces for blood. In other words, taking preventive measures. And we observe that changes in eating habits most likely responsible for dramatic declines in stomach cancer. Very similar complicated relationships among the cancers of women. Happy to say that lung cancer death rates for women have just started to decline, largely because women have begun to practice smoking cessation and in larger numbers than before. Another good example of how science has contributed to control of a disease that, in this case, a disease that wasn't known when we were in college. It appeared 20 years thereafter, namely AIDS caused by a retrovirus, a new retrovirus called HIV. And here, the usual response to the control of a viral infection, the kind of response all of us who grew up in the years of the polio epidemics would have been anticipating, namely the production of a vaccine, has still failed. But the molecular analysis of the genome of HIV allowed us to dissect this virus and its genetic components to identify functions in the virus that were required for the virus to grow, and to develop new drugs that finally have at least partially controlled, not completely, but partially controlled death rates from this virus in this country and increasingly abroad. I point out to you, of course, that transmission of the virus continues with about 40,000 new infections in the U.S. each year. We think that number can become significantly lower if more patients who are still asymptomatic are taking antiretroviral drugs, and that's something you'll be reading about in the paper. But the point here is that the science of the genetics of viruses led to an understanding of this disease at a molecular detail that allowed the creation of new therapies that would not have been conceivable 30 or 40 years ago. I'd like to say a little bit more about cancer. That's what I work on. That's my responsibility these days. I'd like to give you a little bit more sense and perhaps a deeper perspective on how we think about cancer. You all know this is the leading cause of death worldwide. It's number 2 in the U.S. at the moment, but will be number 1 if cardiac death rates continue to decline. It's a disease fundamentally in which DNA has been damaged, mainly by mutations, sometimes by other kinds of chemical changes that occur on DNA. There are, of the roughly 21,000 genes in our chromosomes, there are a few hundred that are especially important in the development of cancer. We think of those mainly as of 2 types, so-called oncogenes, genes that have an enhanced function in a cancer cell and do something more vigorous than they'd normally do, largely as a result of mutations, to propel a cell into abnormal growth and invasiveness. We also have a set of genes called tumor suppressors that normally keep cells from behaving badly, and when those genes are damaged by forces that lead them to be inactivated, then their ability to suppress tumor growth is eliminated. We know the functions of many of these genes, and that is very important. We know a lot about the proteins they make, how they change the growth properties, the invasiveness, the escape of cells from a normal process of cell death. And understanding those functions is critical to the kinds of therapies that are currently envisioned, a few of which I'll tell you about. I've tried to emphasize the idea that every cancer is different, and the only way to understand cancer is to recognize that each cancer type has its own problems, and each individual cancer is going to have a different constellation of mutations, and therefore offer different kinds of opportunities for control by prevention, or early detection, or better diagnosis, or new therapies. I'm going to give you very briefly a few examples: adult leukemia, lung cancer, melanoma, and cervical cancer. But I draw your attention to a book that recently won the Pulitzer Prize by Sid Mukherjee called The Emperor of All Maladies, in which you can find many more stories that are informative and very well told. So chronic myeloid leukemia, a disease of adults occurring in about 6,000 individuals per year in this country and pretty much the same rate throughout the world, is a disease which until just a few years ago was uniformly fatal. It commonly arises in middle age. That is not 70, but more like 40 to 50. It is a disease that has an early, prolonged, so-called chronic phase, and then erupts into a crisis phase, and individuals die shortly thereafter. The key feature to this disease, the clue that has led to remarkable control of this disease, was discovered just shortly before my class graduated from college, when two scientists in Philadelphia identified the atypical chromosome shown on the right, the so-called Philadelphia chromosome. And over the next 20 years or so, this chromosome was shown to be a chimera, a chromosome composed of pieces from two other chromosomes, that happen to be number 9 and 22, that are fused at a certain point that allows an oncogene called the Abel oncogene to make a atypical protein that has a souped-up enzymatic activity that now does something that perhaps I don't want to get into in detail, but it's an enzyme that adds phosphate to a specific kind of amino acid in proteins called tyrosine. And that was an important thing to learn because several years later, a drug company developed a drug that turned out to be an inhibitor of that enzyme. And that drug, officially called imatinib, but commercially sold as Gleevec, remains the poster child for what the molecular revolution in cancer can achieve. This drug blocks that oncogenic enzyme, and it kills cancer cells, and it returns people to normal health. And the most important thing on this slide is that we now know, as a result of use of this drug over the last 12 years or so, that use of this drug and a few very similar agents basically restores a patient who has this diagnosis to a normal life expectancy. And that is a really remarkable finding, not yet duplicated for many other cancers, but it's one that offers tremendous hope.
What is the cancer that this affects?
Sorry?
What is the cancer that this affects?
Chronic myeloid leukemia, the one I'm talking about.
Leukemia.
Yeah. Now there are several other cancers. It turns out that this chemical inhibits at least 3 enzymes of a very similar type, all enzymes that transfer phosphate to tyrosines. And those other tyrosine kinases also have a role in other kinds of cancer, quite rare cancers, but one of them, called gastrointestinal stromal tumor, is actually also a disease that occurs in a few thousand people in the US every year, and this drug is very effective in that disease as well. Lung cancer, the major killer from cancer in the US, causes 1 million deaths globally. As I mentioned, the 1964 Surgeon General's report had a tremendous effect in reducing tobacco use in this country, and that has had a dramatic effect on lung cancer rates and mortality. There are several types of major so-called microscopic types of lung cancer, but now that we know a lot about the specific genetic changes in these cancers, we know that we can subclassify them further. When we do that, we find that there are, again, largely enzymes that are affected by the mutations that can be inhibited by new drugs. And we have at least 2 new kinds of lung cancer, having mutations in certain genes against which drugs are available that can be controlled at least temporarily, not, unfortunately, over the same duration that we control the chronic myeloid leukemia that the Gleevec controls. But this, again, is a harbinger of better things to come. Just this year, or end of last year, we also introduced a new way to try to control this disease by showing, with a long-term study from the Cancer Institute, that screening with what's called helical CT scanning can reduce mortality from lung cancer in smokers by 20% by finding the disease early. This is a problematic new technology. It's one that's going to present serious problems for those who have to supply and pay for health care. But this is an important new modality for screening that will be right up there with mammography as a way to think about this disease. Melanomas arise on the skin, and roughly 70 or 80,000 individuals in this country have a melanoma each year. About 15% of those individuals go on to have disease that spread to other organs, such as the liver or the brain or other organs around the body. Two, this is a disease that is metastatic melanoma, a lethal disease. We've had no advances against it that are useful clinically for about 30 years. But just this year, two new things. First, a mutation, a change in DNA that's found in about 60% of these individuals can now be counteracted with an inhibitory drug. And virtually all those patients have a profound remission. It's not durable, but it's very dramatic. And there are new drugs coming along that I believe will help to sustain those remissions. Furthermore, a way to manipulate the immune system using an antibody that works against the brakes on the immune system has been shown to be effective, was recently approved just a couple of months ago, and this is an approach that's applicable not only in melanoma, but in other cancers as well. Finally, a very different approach to a disease called carcinoma of the cervix, a disease which has been quite well controlled in this country by using pap smears and detecting abnormalities quite early in the production of the disease. And at this time in this country, only about 3,000 deaths from cervical cancer occur. This was not always the case, and for those of you familiar with the best-selling book by Rebecca Skloot about so-called HeLa cells that were derived from the cervical carcinoma of Henrietta Lacks, you can learn a lot more about the disease from that book. We know that this cancer is caused by a virus that circulates widely in human beings, so-called human papillomaviruses, of which there are many strains, two of which are responsible for about 70% of the cases of this cancer. How do these genes work? These genes work by inactivating that class, two of the important members of that class of genes I described for you before as tumor suppressor genes. The new vaccine blocks infection with this virus and therefore prevents infection and prevents cancer. And this is, in some ways, this is the second effective anti-cancer vaccine. We have another cancer vaccine against hepatitis B virus, but this one has a special connotation for this community because a brother of our classmate Marty Lowy, Doug Lowy, who happens to be the deputy director of the NCI and, of course, an incredibly close associate and friend of mine, played a central role in the development of this vaccine. This vaccine is unlike the kinds of vaccines you're used to, where you just grow some virus and either kill it or don't kill it and then immunize somebody by injecting virus. Instead, this kind of vaccine makes use of the technologies I was describing earlier, namely taking the virus apart, understanding its genes, identifying the gene that makes the major protein that forms the coat of the virus, and then by making that protein in another organism, in this case, cells from an insect, you can take the protein and allow it to reform a virus-like particle, which has the potential to immunize human hosts against infection by the virus. And it's that kind of non-infectious, non-oncogenic vaccine that's been used incredibly effectively in just a couple of years since it's been made by two major companies. And now the potential for protecting our own citizens against this virus infection is, of course, tremendous. But this gives me a chance to segue into another important issue, which is that many of the advances we are making through medical science in this half century are going to have tremendous impact in developing countries. So whereas the cases of cervical cancer in the advanced economies is relatively low, in many parts of the world, the number of cases can be at least tenfold higher. Many of these countries can't afford to do pap smears. And the result is that these are places where this viral vaccine can have an enormous impact on cancer that is often the major killer of women by cancer in countries in Africa and parts of Asia and parts of South America. This is only the tip of the iceberg. There are many other cancers that are caused by infectious agents, including the bacterium called H. pylori that's associated with stomach cancer, several other viruses, and other agents. And in the developing world, roughly 26% of cancers are attributable to infection with various kinds of agents. And we see this as a tremendous opportunity to extend the world's interest in global health to cancer. Throughout the developing world, there are remarkable inequities in life expectancy, and that has impelled much of the interest in global health. It's hard to see the numbers from where you're sitting, but life expectancy, which is in the 70s or even low 80s in many of the advanced economies, falls to levels as low as 30 or 40 years in many countries in sub-Saharan Africa, for example. And these create opportunities for improvements in health that I believe we should all be seizing. I include this as just to alert you to the fact that cartographers are playing a major role here too in showing us some of the inequities in the world. And this is a imaginative way of displaying the distribution of deaths from AIDS in relationship to total population throughout the world. And you can see how dramatic the impact of AIDS has been in relative terms in Africa. So all of this has contributed as the great science, the new ways to control disease, the recognition of disparities in health has brought tremendous amount of additional attention to global health. During the George W. Bush administration, a presidential emergency program for AIDS relief was developed that has increased our standing in this world of global health. President Obama has a global health initiative that is expanding the reach of our global health efforts to many other diseases. The UN has set standards to its Millennium Development Goals and has taken an interest in pursuing non-communicable diseases as well. The many foundations, most especially the Bill and Melinda Gates Foundation, have contributed to these efforts, as have both medical students and faculty at medical schools throughout the country. Now, in the last 10 minutes or so, I'd like to talk about two things. One is I'd like to say a couple of things about the behavioral aspects of science, the way science is done, and not just the accomplishments of science. And then I'd like to spend a little time at the very end talking about what motivates the efforts to do science, especially at times when we live under economic constraints. Science is no longer a lonely activity. All science is based on partnerships and teams. I include a few pictures from my own history of my close relationship with my scientific partner, J. Michael Bishop, and of the kind of teams that he and I assembled over the course of about 20 years. These teams are often multidisciplinary. When I was at Memorial Sloan Kettering and working on lung cancer, those teams were composed of surgeons and pathologists and medical oncologists, statisticians, molecular biologists, and many others. And they are inherently multinational. This happens to be a group of my own laboratory personnel taken when the Gates were in Central Park a few years ago, and you can see these folks come from all over the world. There are ways to portray this doing of science and some of its complications. I draw your attention to one recent book, a novel by Allegra Goodman called Intuition, that based on a real lab in Cambridge, Mass, that shows cancer research being done by a partnership with some of the interpersonal relationships, what should we call them, that occur in such a situation, and some of the dark sides of science, like misconduct and inappropriate governmental intervention. Another way in which science has changed culturally is that there's much greater recognition of the importance of sharing information. Some of this came out of the way in which the Genome Project was conducted, with instantaneous sharing of new sequence information so that labs around the world could work together in a collegial fashion, but also an increasing recognition of our responsibility to those who fund our work, namely you, the public, taxpayers, and an increasing recognition that we depend on each other's work and it shouldn't be sealed up behind subscriptions. And the idea was to build public libraries, as we've done at the NIH, and to carry out our publishing in a more equitable way with a novel business plan that requires funding of the costs of research, the costs of publishing research up front to create so-called open access journals. Some of these journals are in a constellation of journals called Public Library of Science that I was involved in funding and founding. And those of you who want to see what this looks like can proceed from an article that appeared in the front page of the New York Times a couple of days ago, an important article showing that one of the reasons why obesity is such a flagrant problem in this country is probably due to the fact that people are leading more sedentary occupational lives. That was information in an article from a journal called PLOS One. What the Times story does not make clear is you can just go type in that something about that article and see the whole article, full text, for free, something that would not happen in a traditional journal where your screen would say, pay $30 and you can see this article. Now, this is important for transforming the way in which we work, making our products, the products of our ingenuity, available to the public, and allows the information we generate to be put to good social use quite quickly. So, a few more minutes on that address some of the problems that we are facing in the funding of science. Just to go back to some basics. So I remind you that of the precepts of Vannevar Bush, the science advisor to FDR and that later to Truman, who set that framework on which most U.S. science has been conducted. That the federal government largely makes investments in basic research, at least in my domain, and that we expect the extensions of that largely to happen through industry. Vannevar Bush described this in the following way. He said, basic research is the pacemaker of technological progress. New products and new processes do not appear full-grown. They are founded on new principles and new conceptions, which in turn are painstakingly developed by research in the purest realms of science. Now, some of you may remember that in 1960, maybe early 61, C.P. Snow, Lord Snow, appeared on this campus and gave a talk about what is called the two cultures, a lecture that he gave in England in 1959, in which he described two cultures, the literary culture and scientific culture, that he claimed weren't talking to each other and weren't working well. We had a discussion at what used to be called the Phi Psi House after this lecture, and I, who belonged to the literary culture at that time, felt the anxieties of anybody who was being told by an esteemed politician, scientist, and novelist that literary culture was over, that the scientific culture was going to cure the ills of the world. Well, that was simplistic. These divisions are not as deep as C.P. Snow alleged, in my view. But his lecture, when returned to, describes another kind of two cultures within science, the culture of pure science and applied science, and a contrast in which C.P. Snow seemed to be taking the side of the applied scientists. Pure scientists, he wrote, have by and large been dim-witted about engineers and applied science. And he said this was due to a willful elitism that he thought was inherently British and built on class distinctions. They, he wrote, the pure scientists wouldn't recognize that many of the problems of applied science were as intellectually exacting as pure problems, and that many of the solutions were as satisfying and beautiful. Now, I don't deeply disagree with C.P. Snow about this, but I do disagree with a notion that is common in current conversations about the expectations the public and the Congress ought to have about funding for NIH and other science agencies. Namely, a notion that our science agencies are engines of economic development with an overemphasis on application of findings, on something we call innovation, which means making something that's commercially attractive, on medical translation, taking what we already know and creating new things. There is a danger in this, and that's the danger of neglecting the process of discovery that we need so desperately, because we only understand 2% of the human genome. We know nothing about the brain. We haven't begun to understand the full complexity of the immune system. Many infectious diseases are still poorly understood. We're just starting to sequence the genomes of tens of different kinds of cancer. And that kind of discovery promises a great deal more in the long run. I'd like to conclude with a reconciliation that comes from the buried writings of a great geneticist, Sal Valoria, who was the head of the first cancer center at MIT. And Loria had a neat metaphor for what I'm trying to say. He wrote, under the pressure of utilitarian society, the cathedral of science has come to look like one of those monasteries that one sees in the French countryside, in which a modest church is almost hidden by a prosperous distillery. The sale of products becomes the justification for being allowed to pray to the Lord. So we need to convince Congress that working in the realm of the purest science, as Vannevar Bush once argued, should not be excluded as we rush to apply the many things that medical science has delivered to the world in the 50 years since I and my classmates left Amherst. Thanks very much. Happy to take some questions for the time that remains. I see some hands down here. Yes, Robert.
I've helped design open access for a large study that shares information broadly in the way you described. But I've gotten tremendous pushback from journal editors who don't really want to give authorship credit to individuals in the group. We have 120 people, and there is a mechanism by which we have an asterisk and we say these are non-writing investigators associated. So my question really is, still at this point in time, the promotion structure doesn't really favor large groups contributing to clinical research. How can we change that?
Let me just say, I think your question is confounding two issues. One issue is the issue of open access publication. And what you're asking about is how do different members of a group all get adequate credit for what they do? That's a different question. It's an important one, but it applies to both traditional and open access publication. I think the way we answer that is we have to change fundamentally the way we evaluate each other for promotion and appointment in academic institutions and for grants at the NIH and elsewhere. Not by citing publications, many of which may have long lists of authors and uncertain attribution of credit to the individuals who's on paper. The traditional CV should go away. We should be writing bio-sketches in which people have a chance to say what they have done, what contributions they've made to work. What ideas did they contribute? What part of this project did they carry out? And what were the consequences of doing so? And I think that is a very important question, because there's another side of the issue you raised that your question touches on, and that is, how do we establish new open access journals, or any journal that has a new wave of approach to publication in the presence of the traditional standard journals like Nature, Science, and others that have so much clout in the academic process. And there is a whole other sad saga of how people are compelled to publish in certain kinds of journals to be accredited in the academic sector. I'm happy to talk about that, but a little off the topic. I'll take some other questions.
We have been asked in our family by your research to submit pathology on every generation
you speak a little louder? I think people can't hear you.
Yale Research Center of Cancer has requested our family to submit pathology because 4 sisters have breast cancer in the last 20 to 16 years. We've all been diagnosed with it. We've all had operations, etc. They would like us, we have indeed given all of the pathological information to them. They have gotten together their own research and advise us that they can find no commonality in our cancer at all yet. But they would like to continue to research within all of the undiagnosed, undiscovered DNA to see if in future they can find any new kind of gene that pertains to all of us. Their advice, and I'd like to know how you feel about this, and I think it's generally interesting. Their advice was for all 4 of us to definitely bank our DNA, which is a simple blood test sent to a laboratory, not at Yale, but nationwide, so that in future generations, our grandchildren, if diagnosed with something, they can do a more pertinent diagnosis. If they can go back to the other generations.
I don't want to step into your family situation here, but I would just make one point, which is that full analysis of DNA is possible now and can be done over the course of the next few years very, very cheaply. So I don't think your DNA will be sitting for generations in a freezer before it's analyzed fully if somebody wants to study your family history. It's now possible, as I was indicating, to do a full analysis quite cheaply, and things will be changing very rapidly. But you're describing a very common problem because cancer is a common disease. There are many families with what seems like an atypical incidence of cancer or multiple kinds of cancer, and those families are very important in trying to sort out the origins of cancer. Why?
I've been very impressed with what seems to be, I don't understand it myself, I'm a complete layman. I have the idea, I have the impression that knowledge of cancer and its causes has advanced dramatically in the last 30 years, but that the actual cure of the disease has not advanced nearly as rapidly as the understanding of its causes and so forth. When do you think that, A, is this correct? B, when do you think that the ability to cure cancer is going to catch up with the advances in the knowledge of cancer and its causes?
Okay, well, I hear this very often, but the point I was trying to make is that every cancer has to be looked at differently. We've made incredible progress against some, much less against others, such as pancreatic cancer. Overall, death rates are declining. The answer to control of cancer is not always cure. Sometimes it's prevention. Sometimes it's just early detection that allows you to cure the disease with conventional methods, for example, surgery. So take ovarian cancer as an example. If we had a way to detect ovarian cancer early enough, we could probably cure many more cases than we do now. So the approach to every cancer has to be structured differently. And it just isn't right to say, when are we going to have a cure? Because there is going to be no one simple solution to a disease, which is not one disease. It's many different diseases. Yeah.
Excuse me.
Sorry,
We
gotta stop.
Yes, actually, we do have a program coming in at
All right.
4:30.
So
if
we'll
you have
get out of
questions,
here,
if you wish
and
to
I'll
just
be in the
speak
back if you
up.
have questions.